So we have begun. All right, and this is, I'm, I'm sure I will have to go over this more than once. This is the most complicated material that we start to deal with. This is the adaptive defense system, and and there's a lot to it. So and I'm doing this identical unit with my advanced physiology class, and the hardest part for me, and I framed it the same way for the group before you, is trying to decide how much detail to include. You got to understand a bit about how it works, but I can tell you in test after test, this is the area that just clobbers. Even these are the advanced phys students. A lot of them are pre-med, bio major students. It clobbers them too. So it's not for whatever reason. This is just a difficult system to comprehend. So I'm going to try to. I've got to teach it to you, and then I will really endeavor to make sure that you know how bad, so to speak, it's going to be on the test part. How what degree of rigor you're going to have in the test. So you have to, and to that end, I will. I'm going to. I didn't send a study guide out for this one yet, did I? You know, I'll send out study guides for this exam and for my micro students exam 3 and that. I figured out give me a project a weekend. My wife doesn't have me doing 16 things, which she does. This is if it gets past. This is the way to frame it. If it gets past the outer barriers, skin, mucous membranes, if it gets past those, and it gets past all those cellular defenses and chemical defenses and inflammatory changes, now we got a problem. Somehow it's going to get into the lymph system, and it's going to get to a lymph node, or it's going to get into the bloodstream, or it's going to get into a cell, and it's going to be discovered. That's this portion of the defense we call the adaptive defense. It is very, very specific. It doesn't. The fact that it's so specific is both good and bad. Okay, the specific nature of it. Let's say if it's a cell that's been damaged, it has a bacteria in it or a virus in it. The cells that are going to be produced, which you'll come to know, are called killer T cells or cytotoxic T cells, will attack just this cell. The difficulty is the immune system sometimes doesn't necessarily obey all the rules. Sometimes there'll be some little aspect of it, and all of a sudden we're going to have these cells might start attacking healthy structures, normal structures. When that happens, when these cells or even sometimes antibodies get out of control, we attack otherwise normal structures. This is the basis for what autoimmune diseases are. Of all the systems, and I had several students ask questions in the previous group, you know, why I have to get all these vaccines over again. They didn't work. It's one of those systems where there's a lot of times we simply don't understand why something worked for one person and it didn't work for another person. Why someone had an irregular, let's say a typical immunization that you all might have received. Why does somebody have an oddball response? We know statistically when someone gets an immunization, there's a certain number of people who unfortunately get sick from it. There's some really bad diseases, something called Guillain-Barré syndrome, which is very nasty. There are some things that are life-threatening. Why they get it, why they don't, we don't know. So a lot of times it's very hard. We understand that in medicine and the folks, whether it's the CDC or other arms, they understand that anytime you're doing something that works with the immune system, there's going to be some fallout potentially. You try to minimize it. Maybe that's the best way to frame understanding that I'll give you what we consider are mostly the rules, but there's a lot of exceptions to these rules. It just doesn't work the way you want it to all the time. So here we go. So resistance. Now this line is how we mount a battle, a resistance against pathogens or their toxins or metabolic byproducts. And it all depends again on whether they are inside a cell and altering the cell's markers. You're going to come to know that these are MHC, major histocompatibility complex markers. And years ago, we would refer to these, and some still do. Certain tests are done on what are called histocompatibility, or HLA, which stands for human leukocyte antigens. Years ago, when I was telling the other class, these HLAs, before we knew a lot about self and non-self, before we knew about some of the things that you're going to see in this presentation, some of the very sophisticated detail that you're going to see in this presentation, we knew about these markers. And I told the other class I had the privilege of working back in 1976 with a doctor who was writing for medical journals in 1936. He was a man probably my age then, and this is 40 years ago, nearly 50 years ago. He was a rheumatologist, and he had written studies about how these markers had impacted and caused certain kinds of, apparently were immune disorders, which were rheumatological disorders, different kinds of arthritis that were based on the immune system. And he was a rheumatologist, so he was an expert at this. And I would sit there and gobble up this stuff like it was going crazy. We were working in a vet's hospital, and I would sit next to him, and we'd see patients. And then when the patients would leave, we'd be writing his notes, and he'd be going, "I did this. What do you think about this?" Well, and it was just like the ultimate learning experience to work with some of these folks who had so much experience. But long story short, we've known that the immune system has been a culprit. We're just starting to understand it. Some of these things are things that I didn't get 50 years ago when I was learning about how the immune system works, just to give you the idea of how much advancements we've made. So it's all about self or non-self. If you did not know, and part of what I was trying to say before is that for the first 6 months of our life, you get from your mom antibodies. That's your protection. Your immune system is just starting to function, but hasn't really ramped up to speed till a minimum of 6 months old. I mean, and you'll find that with schedules for immunizations, they don't start till about then, because they wouldn't be of any value, to tell you the truth. Because they don't start doing them until they feel that you can an immunological response, whatever you're feeling about. Is that too early or not? There's still always going to be subjects of controversy with regard to that are there. And again, that's when we start to distinguish. At that point in time, your immune system is figuring out what's self. And it's not a visual thing. It's really more of a chemical thing. Your white blood cells that are starting to form and are basically communicating with other white blood cells, they're going over structures. All cells in your body have little markers that are external, part of these MHCs, okay, that protrude. And on the ends of them, they have different molecules. They have sugars. Sometimes they'll have fat. Sometimes they'll have short peptide chains. Those are all the things that distinguish self and non-self. So, I mean, and it's a structural thing. So these white blood cells will roll over them, and as they roll over, they're able to identify, oh, that should be here. Oh, that's normal. That's normal. I'm getting used to you. So whenever in the future, any of my brethren, any of my other white blood cells that your body produces in the bone marrow are going to say, okay, I recognize what you are. And if all of a sudden one of those cells is altered, or one of these products is floating around in the bloodstream, or one of these products is sitting on the cell that has a foreign marker, let's say if it's used to this and all of a sudden it gets this, the minute it rolls over that structure, it latches onto it and triggers a very, very vigorous response. Vigorous doesn't even do it justice. A very powerful response. So once your immune system is activated, it causes a lot of problems. One of the differences when you and I produce antibodies, if we had that cold or that stomach bug, antibodies thankfully have a lifespan. Cells only produce them for so long and they run out of steam. So we don't have to worry unless all of a sudden it's some kind of a tumorous cell that's making antibodies, and they're not that common, to continually produce antibodies and cause problems. They're sort of self-limiting. They're there. Hopefully by that time you get better. If you don't, that's a whole other issue that's there. Aspects of this aren't quite like that. A lot of these immunological cells and their components keep working until they're told to stop. That's potentially a big problem. That's another way, one of these things where we see immunological disorders. It's almost like the stop signs are missing. You'll see a lot about these things. And sometimes there's too many stop signs. What happens with HIV, when it becomes AIDS, is basically the ability for those stop signs to work disappears. I'll comment a little bit more on that. Cells that trigger the immune system don't work particularly well, and the ones that shut down the immune system almost overwhelm the good ones. Would be maybe a way to express it. And at that point, it makes the transition from someone being HIV positive, which is by no means, which is by not by any stretch of the imagination fatal, to a condition called AIDS, when their immune system has collapsed, and by definition, it's fatal. And what does fatal mean? You're going to die sooner than you should. That's what fatal means. Doesn't give you time for it. Typically, if it's untreated, it takes 2 to 10 years. It's a relatively slow process. Some people faster. And again, there's variations in everything. Some people, depending on their overall health and their exposure to things like that, get sicker faster. And I mean, you get some people in that 6 months to 2 years span. We call that, it's like a fulminating condition. And there's exceptions to every rule when it comes to living systems. So years ago, we used the terminology for all of these things, antigens. Antigens are not, and this is incorrect, are not non-self molecules. And you'll see subsequently they say foreign antigens or non-self antigens. Those are foreign. All of our cells have antigens. They're self versus non-self. That's that recognition period. So antigens, originally when I learned about the immune system, everything foreign was called an antigen because it meant antibody. Here's the derivation: antibody generating. We used to think the way your immune system worked was strictly through antibodies. To understand it, in the last 50 years, there's been a lot of changes. That's when I was learning about it almost exactly 50 years ago. So antigens, now we realize they're self and non-self antigens. Okay, and so this is what evokes an immunological response, something that's either completely foreign or even a marker that should be there is altered by some process. Like a cancer cell would be an example of that. So they're carried out by the particular white blood cells we call lymphocytes, or the arm of this. Macrophages, similarly, they play a really important role. Sometimes they activate. More often than not, the role that they play is effectively gobbling up the debris. If the lymphocytes are killing or disabling or immobilizing the bad guys, be they cells or viruses or bacteria, they're just by themselves, or other things, what happens is once they're immobilized, basically white blood cells come on, one called macrophages, which means big eaters, and they gobble up the debris and get rid of it. That's the way that they work. So we have 2 types of these: the cellular one, okay, which is, and this is where you see over there that I've written cytotoxic, or T sub C. We sometimes erroneously, it's not the proper name, but it's the easy way to remember. They're called killer T cells. And how they work, basically they attach onto foreign cells or cells that have been invaded, where the markers have been altered, and destroy them directly. It's almost like they shoot chemicals at them that cause them to burst, destroy them, whatever way. Not thunder, is it? It's just noise.
I think it's there.
Oh, good. I forgot we're close to the airport. That would be logical. Stop. And the humoral means a body fluid. Antibodies are found in body fluids. Antibodies are found in the plasma. And antibodies are found in tissue. So they are a fluid arm, and that's a different arm. That arm, I think, is a little easier to understand, particularly the different categories. That's one of the things that I find students do not have a lot of trouble understanding compared to this. So once it gets through those outer barriers, and now the second line defenses, now it's going to activate one of these third line defenses. And the specificity is important. The fact that you found antibodies to that cold doesn't mean that the antibodies to the next cold or that flu bug or that stomach virus are, because it's totally specific. And that's why there's so many different cold viruses. There's hundreds of them. And they have a bad habit, unfortunately, of mutating anyway. And that's why people talked about, why is there no vaccine for a cold? Well, the reality is twofold. One, colds are not deadly. So, and vaccines can cause people to die. I mean, there's clearly a risk factor with immunizations. And two, it's almost impossible because of the rate of mutation to justify making one. It's a lot. I mean, so a lot of that goes into it in regard there. So antigens. So there are specific receptors on these traveling white blood cells that are there to recognize self, and now they're saying it appropriately, non-self. The system responds to non-self, but not to self normally. That's a big deal when it comes to these cells. These lymphocytes that circulate, what they have when their process of maturation involves being able to recognize and not react to things that are self. They have to be able to recognize the things that are self and not attack them, and recognize things that are foreign and attack them. So there's a lot that goes on, particularly with the line of T cells, as we will see shortly. These foreign markers, proteins, polysaccharides, so they can be, relatively, polysaccharides are longer chains of carbohydrates. Glycoproteins are probably the most common. Those are little short chains of sugars that produce from proteins that are embedded in membranes. Even fats have sugars attached to them called glycolipids. Okay, and typically, the larger they are and the more complex they are, they're pretty problematic. This is an important term. Hapten was derived from the term half antigen. This is where allergies come from. Allergies or allergens in and of themselves are not enough to provoke an immune response. So let's say, for example, cat dander or dog dander, tiny, tiny particles. When we inhale them, they contact other proteins complex with them, and effectively that's when the allergic response comes from. By themselves, they're too small to generate an antigenic response. So they combine with these larger molecules, typically a protein, and that evokes the immune response. So, and we do a little nugget on allergies at I use a different PowerPoint, and I'll go into a little bit more about different kinds of allergic responses. Allergies are pretty common, as you know, and a lot of times these tiny bits of pollen. We're in the time of year, I'm getting it now a lot. When spring, I have spring and fall allergies, which are very, very common. These are pollens and different things. A lot of people that are there. Some are more severe than others, to be very fair. So lymphocytes start being produced during fetal development. All white blood cells are born in the bone marrow. They don't all mature in the bone marrow. Lymphocytes particularly. They are born in the bone marrow. About half of them eventually get delivered, as you will see, to the thymus. So here's how it goes. Red bone marrow releases these effectively white blood cells that really don't necessarily have an identity, and typically in their precursor form. Although half of them settle in the thymus, half of them do not become T cells. T cells are the most aggressive of all the white blood cells. They release the most chemicals. They're most often responsible for autoimmune disease. Only about 2% of that 50%. So a relatively small number of those. And they go through something which we're not going to go into. And I glossed over it with the advanced class, called negative and positive selection. So that's all that identifying things that are self and not reacting to it, not bonding to things that are non-self and then attacking it and making sure you attack it if it's necessary to attack it. All that type of stuff is this portion of this positive and negative selection. What happens to the other 98% of those T cells? They basically undergo apoptosis. They basically, because they have a lifespan, they deteriorate. They either stop being made, or they undergo apoptosis and are destroyed, or they just basically die because they have reached a certain point at the end of their relatively limited lifespan that are there. So the rest of them stay in the bone marrow, and they are, because they are in the bone marrow, we refer to them as B cells. This is the origin of the B cell term, this bursa of Fabricius in chickens. I guess that's where it was studied. We never learned the term. We called them B cells because they were born in the bone marrow, raised in the, matured in the bone marrow and made antibodies with a B. T cells were all about the thymus. Whatever works for you is fine. What you're going to see that it's an easy way to remember as far as I think of. So when you look at a blood smear, if you did that in lab, or if you see a tissue smear and you see lymphocytes, most of them are T cells. A limited number are B cells. B cells, when they're active, change into something called a plasma cell. And the best way to describe a plasma cell is kind of like a factory that makes antibodies. It changes. It has a larger amount of rough endoplasmic reticulum. That's where proteins are made. And it becomes a mega factory for this. It has a lifespan of maybe 5-6 days and then basically dies. And those antibodies circulate until they finally are no longer available. That's what I mean. There's a lifespan, thankfully, with antibodies, so you don't have overproduction problems who are there. And what they do, once they mature, then they go to the usual places where we encounter problems. Okay, for instance, lymph nodes, spleen, okay, the thoracic duct, some of the structures we talked about in the lymphatic system when we looked at the anatomy, white pulp of the spleen, staying in the bone marrow. Typically, the intestinal lining is more often a parking place for B cells, okay, is because typically what we encounter when we ingest, the thing that causes most problems are toxins. So food poisoning, which you and I call food poisoning, is really, when we look at this, or I'll look at it with my micro students, it's sometimes called a food intoxication, because you're responding to a toxin. It's there. Food poisoning and food infection are not the same. There's only one type of food poisoning that's immediately lethal, and that's botulism. Okay, when you get a dose of food poisoning, you're sick for 24 to 48 hours, vomiting, diarrhea, feel like awful, and a couple of days later, you're better. You rehydrate, etc. That type of stuff. It's not a big problem. Food infection causes damage and get into your bloodstream. And the end product could be, if it was something like a bad salmonella or E. coli infection, could shut down your kidneys and put you into ICU and be close to killing you. It happened to somebody close to you, me, back in 2006, almost 19 years ago. I ended up with a food infection that ended me up for a lovely 3-day stay in ICU in St. Margaret's. Not fun. How sick was I? They called my 87-year-old mom, who was alive at the time, and told her I was close. And her 55-year-old son was close. So I was a sick puppy. I know, smoking those 2 packs of cigarettes every day was not really a good idea. You got to cut down. And you're really in trouble. We're not even talking about you. Not you, Grace, because you wouldn't do that. You're healthy. You have to make, if I don't at least have to tease Grace once each class, I feel like I'm not doing my job. Moving on. So, I don't get a whole lot out of this. It's in the bone marrow, it's in the thymus, it's in the lymph nodes. Fine. Good idea. So, so these are the kinds of things that it does. So, it provides the cellular-based immune response. T cells interact with directly cells that have been damaged and destroy them, versus the humoral or fluid arm, where antibodies circulating chemicals do that. Antibodies don't destroy bacteria or viruses. They immobilize them, they neutralize them, they slow them down, they make them more visible, they cause big clumps of them to form so that macrophages, white blood cells, can come along and gobble them up and get rid of them. That's their job. So a lymphocyte has to be activated before it can respond. So that requires something called an antigen-presenting cells. We have a lot of these. Some of them are B cells themselves that are antigen, but most of the ones you heard about in the skin, there was a cell called a Langerhans cell. Basically, it's sampling the environment. We have them in the liver. We have antigen-presenting cells in the spleen. We have them in lymph nodes. These are cells that latch onto, and the way they do it is they latch on to potentially an invader. They gobble it up, and what basically they do is when they call it presenting cells, they present the little part that's foreign. So they're like the little tip of this marker is that foreign entity. That's what your body, you make antibodies to attach to that, specifically to that marker. It won't fit any other marker. So if I had a red one like this, it would be the same, so it wouldn't attach. It would be similar, but it would be different. Okay? That marker goes by the name either a determinant, as classically the name they use now. We used to use the term epitope. Maybe they use that more with allergy. I don't know. So what happens again, there's these specialized white blood cells called antigen-presenting cells, and some of them are B cells, some of them are the cells that we see located. Basically called the Kupffer cell, KUPFER in the liver, Langerhans cells, we learned about the skin, and they have one of the four epidermal cells that are there. Like I said, they latch onto it, they gobble it up, and the only thing that they present to lymphocytes is that little marker, because that's everything is going to be based on that structure to attach to. So it phagocytizes it, gobbles it up, just displays that fat fragment, and it displays it as part of one of these complexes. Okay. There's a difference between them. One is called MHC1, one is called MHC2. I don't want to go crazy with that. Okay? Because that's one of the things that inevitably, when I analyze what questions students get wrong, they get those wrong. That plays a role in this. Those, when it's a T cell or a B cell, when it's finally matured, T cells in the bone marrow, T cells in the thymus, and when they mature, the name that we give them is naive. They don't, once they encounter an antigen, or one of those antigen presenting cells, that starts the process of activating. So mature cell is naive, that means it hasn't been exposed to an antigen. Once it's exposed to an antigen, now it becomes activated. In the land of T cells, there are two types. They have different markers. When they're in the naive form, they're called CD4 and CD8. When they're activated, they will either become, CD4 will become the single most important cell in your immune system, the helper T cell, or T sub H, or you'll become the cytotoxic or killer T cell, T sub C. That's why that is such an important cell, and it tells a tale about HIV/AIDS, why it is so pervasive when it's not treated properly or inadequately or not at all. What does its target? The helper T cell. So it destroys your helper T cell line to the point, and now the definition of when we make the transition, someone who's not undertreated or not treated, very ill to start, makes the transition from HIV positive to AIDS, their helper T cell population goes below a critical level, and at that point their immune system collapses. And there's no way back. It's not like magically you can get it back. Not that we know of now. There's, we're seeing studies that are showing people who may have survived or for X amount of time, but at this point, it's a little bit easier just to say, hey, now at that point they've got AIDS, and that's going to shorten their lifespan or be fatal. So when a specific T cell encounters that antigen presenting cell, okay, it now is activated. And what happens is they begin to secrete. So the arm of these cells is chemicals. The name of these chemicals are cytokines. I talked about them before. Most of the time, they're going to stimulate inflammation, cells to multiply. They're going to reinforce other cells. I mean, one of the things like a helper T cell will do, it sends chemical signals to the innate system. You know, we're dealing with an infection now. You're not doing your job. Tighten up those boundaries. Get a better eye on your surveillance ring. You know, it's again, if somebody got through your front, if somebody got through the front door, that happened to us. You know, where we lived in Cranberry, we never locked our doors like that. Just didn't need to do that. And one time, there was actually, my wife and I had gone to bed. The dog, our former dog, great dog, was in bed with us. Little did we know, my stepdaughter and her boyfriend came back like at midnight. Somebody was in the house and literally snatched her purse. Didn't even see her. This gal who was eventually caught. What did the dog do? Nothing. Wonderful dog. Did he bark? He'd bark at anything else. But when here's this woman who was drug seeking, was seeking drug money in the area. Our dog now would do the same thing. He'd bark, but he showed them where the silverware was. Here, valuables here. He keeps his wallet over here. The car keys are there. Very accommodating dog. But that's basically what your immune system does. What these cytokines would do. Hey, lock the door. You know, you've got a problem there. You, dog, learn to bark. Effectively, if you want to use that silly analogy, that's kind of how this system works. It reinforces the other systems. Activates the other cells by secreting its own cytokines. We'll go into some cytokine specifics, but not today. Stimulates B cells to make antibodies. Stimulates T cells to make these cytotoxic T cells that attack damaged cells. So the cytotoxic cells can be cancerous cells, transplanted cells, transfused cells, cells that are infected not just by viruses, by bacteria. Tuberculosis is a bacteria. Okay. It's attacked by these kinds of cells. Not very effectively, but if it could be, it would be. And of course, these lines generate immunological memory. It doesn't matter whether it's making antibodies or whether it's making cytotoxic T cells. Immunological memory is the foundation of why we use vaccines, or why once we have a particular disease, we have a substantial amount, if not lifelong, immunity to it. As a kid, I got measles and chickenpox and German measles. They're still around. Haven't got them. I did not receive any of those immunizations. It was a long time ago. I had chickenpox and measles when I was 7 years old, 6 or 7 years old. German measles I had when I was in high school. So a pretty long time, and I've already had my 50th reunion. So you do the math. I skipped 55. I only go to ones with a 0 after. I'm terrible. The only reason I go on the website is to see who with us. I figure as long as my name's not there, I'm good. You know, one of my classmate of mine is a retired professor from Washington University in St. Louis. And he kind of took over because nobody was doing anything about the reunions. And he got so mad about it, he says, I'm going to do it. So he got a whole network of people. Nevertheless, that's the idea of memory cells. They can last a lot. Sometimes they don't. Sometimes you need to get booster shots. Tetanus is different. We know that there's a lifespan. You're going to see, by and large, we know that it starts to wane certainly by 10 years, and usually that 5 to 10 year zone is where typically you want to make sure they have another unit of prophylaxis. Again, if you did not know, when you give someone a tetanus shot or you receive a tetanus shot, you're getting 3 immunizations at once. It's called trivalent. Diphtheria, pertussis, and tetanus. I go into that a bit when we talk about micro and immunizations in there as well. And then these are regulatory cells. These are another one that are a big problem. We used to call them suppressor cells. Problem is, if they're not working effectively, this is a source of autoimmune diseases, where because it's not like antibodies. The cells that make antibodies stop making them after a period of time, and they don't last forever. Okay, they effectively have a lifespan, both the cells and the antibodies themselves in your system. The interesting thing about these is these regulatory cells have to shut it down. Okay, so you don't end up with an autoimmune disease. If the shutdown system isn't working properly, it just keeps going. And then you can have accumulation of these cells, and eventually they're going to start to attack healthy structures. One of the theories behind the origin of autoimmune disorders. This is the hard part. Okay, class 1 versus class 2. Class 1s are on all red blood cell doesn't have it. Platelets don't have it because they're really not cells, but all nucleated cells have this class 1. When a bad guy has infected a class 1, has infected a cell and taken up space, it's altered the cell. So whether it's that HIV virus that got in the cell, the tuberculosis bacteria that got into the cell, plague, for instance, enters cells. I mean, just giving you some ideas of ones that that stimulate, or it's a cancer cell, or it's a transplanted cell. That's going to alter these class 1 markers. That's going to basically trigger this arm, cell immune response, and trigger the formation of cytotoxic T cells. That's what you have to know. I'm going to try to figure out a way to say it so that when you see class 1s that are altered, you'll know that. You know, we have to find a way to do that effectively on the exam when the time comes. I'm guessing this exam is going to come probably right a little, either the Monday or the Wednesday before break. We already did. Maybe it'll come a little sooner. No, yeah, this one maybe will be the Wednesday or Friday of the week before break, whatever those days are. Whatever those days are. Anybody help me? The 8th, 10th, maybe like the 12th, 10th and 12th. Maybe it would be a Wednesday. It would be, so 16th. So it'd be 9th and 11th. Good Friday's the 18th, I think.
Yeah.
It's a Jewish guy. What do I know? That's when we play golf that day. So my wife will yell at me. You don't need to yell at me and tell me I say stupid things. She tells me that deal. Again, she has a romantic way of telling me I'm talking out of, well, we've already discussed that. Class 2s, okay, a little bit different, okay. Those receptors are the ones that trigger the helper T cells. That's as much as you really have to know about those. So only these cell types can act as antigen-presenting cells for helper T cells. Helper T cells are important. I mean, I don't think me going into this in any great deal of response. Once a helper T cell sees one of these antigen-presenting cells, then it starts to make this variety of chemical agents. We will go into in more depth. For the time being, this is kind of what they do. I mean, think of what the helper T cell says. We have an invader. Okay, what do we have to do? We're going to need more troops. So the bone marrow is going to need to make more lymphocytes to fight this infection. Some interferons are part of the innate system. These interferons are much more specific. Okay, they are much more aggressive about viral replication and engulfing viruses, stimulating B cells, and even attack cancer cells. When we started to identify interferon, it's a protein. Started to be able to began to make it genetically, and it was really a frontline treatment to treat cancers, different types of cancers, not necessarily that effectively. And before we had better antivirals for hepatitis C, that was one of the treatments for hepatitis C. So interferons are important, and there's different categories of them, just like there are many categories of interleukins. Interleukins, you're going to see, and I'll go over this with a different power point are going to the name ILs. Truly the most important of these is IL-2. IL-2 is the one that's made by helper T cells, and that's the one that reinforces all parts. It stimulates the cell system, it stimulates the antibody system, it tells the innate system you're not doing your job, all of those kinds of things. It's sort of the universal, it's the most powerful of these engines. And then we start to isolate and make interleukin-2 in the laboratory, or refine it in the laboratory, and that was an early treatment for treating cancers. They are very powerful agents, and we'll go into those. Tumor necrosis factor. This is something that, because cancers are part of this, we make tumor necrosis factor the signal when these are to stop tumor growth. Okay, all of these things releases growth factors, etc. Fever that sometimes we'll see part of it, bacterial infections, stimulates lymphocytes, One of the original, I took one of these because I have an immunological arthritis. The idea that something that is designed to suppress cancer, for me to take it, because I didn't feel that great when I took it, to stop an arthritis, which was annoying but not disabling, as it were. And I didn't like the way I felt. I felt very funky. I couldn't even come in and teach. And this wasn't that long ago that I stopped it. I probably stopped this right around the time we first met in the fall. I was taking something called Humira, which you see advertised on TV all the time. That's something that fights tumor necrosis. My first wife took one of these when it's early days called Enbrel, which is still around. There are different kinds of ones. And unfortunately, she ended up developing a tumor. She had a disease that causes tumors, unfortunately, and that probably predisposed her. She has a disease called neurofibromatosis. She has a comparatively mild presentation. One of the major presentations of that you may have heard of is called the Elephant Man disease. She has a minor one that causes neurofibromas, which are kinds of tumors to occur. The Elephant Man was totally disfiguring. She happened to form a tumor inside that turned out, we thought it was a lymphoma, that turned out to be a neurofibroma in her gut that was pressing on, it really caused a bulge there. It was a little bit smaller than a softball. So we were really worried that happened. That was 2004. She unfortunately has had more health problems than, that have just sort of, it's usually one of those one things after another. Goodness. Sunday night she fell out of bed in the apartment that she lives in and clunked her head, ended up in the hospital with a concussion. Oy. Poor daughter. So B cells can be activated based on the shape, and then B cells become more activated by cytokines, particularly from the helper T cells. They can form memory cells, okay? And again, the big player in making antibodies are plasma cells. Plasma cells are like little factories that make antibodies, but we're going to look at them. We're going to name the 5 major types of immunoglobulins. That's usually not that difficult, and the major roles that they play. So, and they really are the humoral response because they travel in the bloodstream or in fluids, and fluids, another name for fluids, are humors. We're stopping here. This is not a particularly good. I have an animation I'm going to show you on Monday. This is about what goes on here. So here's a B cell. The context, let's say it's a virus or a bacteria. It forms antibodies. And then some of them, and these antibodies come from these plasma cells, or as some become memory cells. Thanks a lot. We will see all of you folks on Monday. Have a great week.